Welcome everybody, and thanks for joining us for a new series of evenings at Whitney. Hope you all had an enjoyable summer and that you survived the extreme heat pretty well and it didn't cause you too many problems. It's my pleasure to be introducing you to Professor Elizabeth Clare tonight. If you haven't already guessed, Professor Clare works with bats extensively, but not just bats. So she's primarily works on the taxonomy of bats and their diversity in Central America. But she's also conducted a lot of field work around the globe, including places like Israeli deserts, the African savanna, Costa Rican rainforests. Her main study sites are Costa Rica and Belize in South America, but she also works extensively in Texas and her native Ontario as well. So I think we're in for a bit of a visual treat, given how well-traveled Beth is. And just a little bit about her academic background. She studied for her PhD, and I hopefully won't butcher this, but at the University of Guelph in Canada. After that, she made the move to the UK, where she conducted a number of postdoctoral fellowships, one at the University of Bristol, and then a follow-on one at the University of Cardiff in Wales. And after that, she took up a faculty appointment at Queen Mary University in London, where she stayed for 18 years growing her research group. And then more recently, back in 2001, she moved her lab back to Ontario, and she's now a professor at York University in Toronto. So in addition to all of her bat taxonomy work, and she's also highly internationally recognized as well for applying genetic approaches to study dietary ecology in bats and their species as well. In more recent years, she has moved into creating innovative ways to study terrestrial biodiversity. So I think we all know that biodiversity is under threat globally, and new solutions are urgently needed to be able to study and understand the changes that are happening at pace. And this new approach has garnered her a lot of high profile, not just scientific attention, but also media attention as well. So her new research directions have been very well received. So I'm delighted to say that's the main focus of her talk today, and we're going to hear all about it. So I don't want to steal any thunder. And the title of our talk today is Tackling Global Biodiversity Through Harnessing the Wind. So it should produce for an interesting talk, and I'd just like you to join me in welcoming Beth.
I have to hold the microphone because I haven't got any pockets to put the little devices they give everybody. Thanks for the introduction. I'm really pleased to be back in person giving lectures. It's so much fun. I'm tired of doing this over Zoom, so I'm very pleased to have visited. It's a beautiful location. I've never been to this part of Florida before, and I really enjoyed walking around and looking at your coastline. Yes, so I'm trained as a bat biologist first. So my expertise is in the identification of bats from Central America. That's kind of where I started. But my students and my team that work for me, we work all over the world. So we've been part of some of the largest ecological experiments in the world. This is a picture from the Atlantic forests of Brazil, where we've done a lot of work. I had students who were part of a team of 500 different independent researchers studying deforestation in Malaysia. I have taught field courses all over the world, including this is the Negev Desert in Israel. It's basically the easternmost extent of the Sahara. We have dug up dinosaurs in Alberta. We've studied bat adaptations to deserts, but mostly I work in Central America. So this is part of our research team capturing bats along forest trails in Belize. And I've had the privilege to work on a team there for 17 years. And we go down every year for 2 weeks and we study bats. A whole team of us go up to 60 people at a time to try and understand one population of bats over time. And I always show at least one picture with bats in it to convince you they're not all little black or brown things. This is the 9 different families of bats that I would catch in Central America. They come in every color, in every morphology. They occupy virtually every ecological role, and which is why they're so interesting to study, because they are one of the most diverse groups of mammals you can find. If there is an ecological question, there's probably a bat that does it to study. So they're a lovely group to look at that. But beyond my work on bats, where I spent my PhD basically pointing out that half the world's species of bats have probably never been recognized by science. That was the conclusion of my PhD, so we've missed half of them. Every one is a cryptic species. I'm part of a much larger group, and my supervisor of my PhD was a man named Paul Hébert, and he invented something called DNA barcoding, the use of short stretches of DNA to understand species. So we can use these little things like forensic scientists to identify whether it's this bat or that bat. And the current global project we're all working under is something called Bioscan. So I'm one of the primary investigators of Bioscan. This is a global project. We're trying to create genetic libraries for all of life. And that is not a simple task, but we have created tremendous public resources. This was the map of where our samples have been collected this week when I looked it up. So this is one global repository. There are 14 million DNA records in that repository from 350,000 species. And that has a very tiny portion of the diversity that's out there. But this is a truly global effort to create effectively a genetic map of what biodiversity looks like. And it's interesting, we're all here because we are interested in animals and biodiversity and conservation, but I got asked a question on the radio recently, which is, why is biodiversity important? And if you work in biodiversity science or you're interested in it, it's so intuitively obvious that I couldn't answer it. I was struggling to explain, and I gave some sort of story about bees and pollination. And so for the purpose of this and for a grant I had to write, I sat down and made a list. So let's start with food security. You don't get any food without biodiversity. There's tremendous amount of evidence to show that everything from the soil biodiversity impacting the flavor of your favorite wine, to the productivity of crops, to not just the bees, but all the other invertebrates and mammals and birds that do pollination, that if you do not have a good biodiversity, you are threatening your food security. That's number one. Number two is water purity. Biodiversity is chiefly responsible for filtering your water to make it drinkable. And living in a coastal area like this, it is very important. It's filtering out contaminants, it's filtering out pollutants from your water. So better biodiversity means more efficient filtering and better water quality. It has a huge impact on regulating climate. There's really interesting studies going on right now in Montreal, in Quebec and Canada, and Toronto, looking at the age and species of tree in people's backyards and local temperature schemes. And there's good evidence that increasing the diversity of trees can lower the temperature in a city by 4 to 5 degrees. Now, there's an interesting way of countering warming climate, is increase your tree coverage. This is actually a graph of rural, commercial, and urban and heat maps. And if you increase the diversity of the vegetation in a city, you can draw that temperature down. Erosion, local biodiversity is really important to preventing erosion. Really interesting experiment done in the national parks when wolves are reintroduced in Yosemite, having a massive effect on regulating biodiversity. And one of the net effects was a reduction in erosion on streams just by top-down control of biodiversity. So a huge impact on erosion. One of the first places we look for new pharmaceuticals and drug discovery is prospecting in biorepositories. Culture. We have increasing emphasis in many countries, including Canada, on working with Indigenous populations to learn about biodiversity. And I've become more and more aware of the cultural practices based around Indigenous knowledge keepers who can tell you about changing biodiversity patterns in highly culturally specific groups. Art, tremendous influence on artistic expression. Human health, lots and lots of links between exposure to green spaces and better biodiversity and human health effects and mental health effects. And to the point where there's a report from the United Nations on the direct connection between greener urban cities and better mental health. So biodiversity is important for a vast number of reasons. And I haven't even gotten to the economy. The estimate of the contribution of better biodiversity to economic output is measured in the trillions of GDP dollars. But given that problem or that benefit that biodiversity gives us, it's always shocking to describe the decline of biodiversity. This is a report that The Guardian put together a few years ago, that since the rise of human civilization, the number of individual, not species, but individual mammals has been reduced by up to 83% in some groups. So roughly 83% of wild mammals have disappeared, 80% of marine mammals, about 50% of the plants, and 15% of the individual fish have gone. And what's the replacement? If you actually did a survey of what's out there, there's about 6,500 known species of mammal, but 96% of the individual biomass is livestock. So humans, cows, chickens, and pigs account for 96% of the biomass of mammals. Only 4% is wildlife, because we are totally dominated by cattle and us in terms of biomass. Same thing for birds. There are 13,000 known species of bird, and 70% of the individuals are poultry, chickens. So chickens make up the vast majority of all known bird individuals. Okay? So this is the problem of biodiversity. The first problem I'll talk about is that we have completely swapped the skew of what's out there. Okay, the next biodiversity problem that most people don't realize is that roughly between 90 and 95% of species living on Earth now are totally unknown to science. We don't know what they are. Half the bats we're not sure about, and those are mammals you can see and they're big. Extrapolate that down to insects and soil nematodes, and one of my friends says the tiny little things that make ecosystems function and provide all those services like pollination and water filtration and soil biodiversity, we don't know most of them. And the reason we don't know most of them is that we haven't got any infrastructure for monitoring biodiversity. We have plenty of international legally binding agreements that ask us to monitor biodiversity, the Convention on Biological Diversity, the international trade agreements, ask countries to monitor their own biodiversity, but we can't actually do it. Nobody can do it. 90% of the species living on Earth are totally unknown to science. And we don't have the infrastructure to monitor it. And so all of those reports about declines are actually guesses. What we do is we do what I do. You go down to Belize and you measure bats for 2 weeks every year over and over again, and you get some idea of what's happening to the bats in one location. You then use that to extrapolate what's happening across Belize, and then extrapolate to other mammals, and then try to compare what we do to what someone else does. And you end up with these sort of very taxon-specific or highly local measurements, and then you try to make sense of that data. The problem with biodiversity is nothing can be standardized. It's incredibly labor-intensive to do. You need someone like me who is trained to identify the bats to go and do it. All biodiversity requires different approaches. Okay, so segue over then to this stuff called environmental DNA. So what's environmental DNA? If I came up to you and I swabbed your cheek and took a sample, I could sequence your genome. Well, I could get David to sequence your genome. That is a direct sample. But there's other DNA. If you all left the room and I came in, I could swab your seats, and I could also get a sample of DNA. We are all shedding DNA all the time. All individuals of all species and all plants, all mammals, all birds are all doing it all the time. It's dead skin cells. It's saliva. It's broken off bits of hair. It is degrading material that just sort of ends up out into the environment. This is called environmental DNA. So we call it eDNA for short, and it's not a new thing. It was discovered in the late 90s by Eske Willeslav in the UK. He's a hilarious guy. I've had the pleasure of doing some interviews with him a couple of times. He found it in ice cores and sediment samples, and he discovered that going back 100,000 years in ice cores, he could find DNA of extinct animals. He found mammoths and caribou and extinct moas in sediment. And he was able to reconstruct basically ancient ecosystems from DNA left behind by the animals long after they'd gone. Now, since that very early work, it's become really a tool in aquatic biology. This is where it's gotten most famous. This is one of my students, this is Alejandro from Mexico, and he's out trying to find out whether the axolotl still exists in the wild using eDNA. And this is where eDNA is best known. You can take a scoop of water from a river and get a pretty good idea of what species live there. It's made its way into lots of regulatory practices. There are industries based around commercial analysis of water samples for the DNA of aquatic populations. This is a thing we do. But eDNA is absolutely everywhere. ESCA first found it on land. It's known you can take a bunch of soil and get eDNA out of the soil. Honey, if you take honey and put it through one of our magic DNA sequencing machines, you can get plant DNA and insect DNA. You can work out who made the honey out of what from the honey. You can collect rainwater and get DNA washed off things. You can find it in the blood inside a mosquito or a midge or something that is parasitizing you. You can find it in footprints left in the snow. And that's probably the best metaphor. eDNA is like a footprint. You leave it behind wherever you go. Real recently, surfaces of leaves, people wash leaves to figure out what DNA is settled on them. A really neat study I saw recently, someone took a bunch of insects from the forest and discovered that their backs were covered in the DNA of all the other things in the forest. They accumulated on their backs. Even in organic things like rock and coal are good sources of DNA from other stuff in the environment. On land, what we've mostly done is used other animals to help us collect it. So these are terrestrial leeches from Malaysia. If you go out and stand around in leaf litter in Malaysia, they will find you. They're very easy to collect. Now, if you are my graduate student named Rosie Drinkwater and I send you out to go collect terrestrial leeches, they're a lot more efficient than trying to find mammals, get 1,000 leeches together. They've fed on the mammals, get the DNA out of their gut, and you now have an army of field assistants out surveying the mammals for you. And these are all things that she identified in the gut of the tiger leech, including pangolins, the most trafficked animal in the world, in the bottom of the middle. Had I sent her out to find me a pangolin, no chance she'd ever have done it. I sent her out to find me a leech and she finds a pangolin inside it. She got the idea of using dung beetles. These are those famous things that roll mammal dung around. And of course, when they're rolling mammal dung around, they get the DNA of the mammals on them. Dung beetles are even easier to capture. Okay, so that's a dung beetle trap in the top left. Basically, you find a friend to produce a sample for you, and you hang it over a pit, and the dung beetles come in and fall in the pit. And then if you're Rosie, you come the next day and you scoop them out, you take them home, you swab them for mammal DNA, and they're covered in it. She found things like banded civets, another extremely rare mammal. But the beetles were out interacting in the forest with the mammals. And so she was able to do a survey of mammals, having never seen a mammal. Cool, but totally indirect. So you're relying on the leeches and the dung beetles to find them for you. Now take a deep breath. I play this game with kids when I do this. So if you're comfortable with it, take a deep breath. Okay. And at the end, you're never going to think about that the same way again. 3 years ago, I got commissioned by an arm of the British government to write a report. The UK government is very forward thinking in using DNA in conservation and ecosystem resilience. So they commissioned 5 reports in a big thing called Understanding Ecosystems and Resilience Using DNA. And I was asked to write about the terrestrial land environment. How can we use DNA as part of our 25-year plan to maintain and improve our biodiversity? And so I wrote about all these different great sources, leeches and dung beetles and all these kind of fun places you can find DNA. Spider webs. You can collect a spider web and it's full of DNA. Cool. And so I'm writing about all these different places you could collect DNA so you could use it to study biodiversity. And I said, just like the aquatic people use water, we could also try air. Now, if you're writing a report for the government, they make you find case studies to back up the kind of things you speculate about. And I found all kinds of great case studies, except none about air. Turns out no one had done it. The only work we could find was a lot of people saying we should look in the air. It was a theoretical thing. Someone should go look there. There were a few studies looking at plants, mostly for things like pollens, for allergens. And there was a little bit of work on fungus. And beyond that, the only sources of what people look for in the air were pathogens, your bacteria and your viruses. And in a post-pandemic COVID world, we all know about that. That's what people look for in air samples. And so around the time I published that report for the government, my university offered a small amount of funding for what they called potentially high risk, but if it works, transformative ideas. Translate into, we will give some funding if your idea is just so crazy enough it might work. Now, when you go to a funding body and you tell them you'd like to vacuum the sky, they look at you like you're a little bit crazy. But you have to be a little bit crazy sometimes. So I went to them, I said, I'd like some funding to try and filter DNA out of the air. And the first question they said was, why? I said, well, I think this is good. I think this could work, and it works in the water, so why not? And so they gave us some funding to try. And where we started with was these guys. These are naked mole rats. And we just happened to have colonies of them in one of our labs. And I thought, well, if we're ever going to be successful, let's start with the most obvious place we could look, a group of highly social, tightly packed animals in a small space. So we took air samples from inside the naked mole rat colony and out in the room, and we compared them. And there was DNA everywhere. There's naked mole rat DNA in the room, and there was people DNA in the naked mole rat tunnels, and it was really cool. So then we went outside, because the only way this would be useful would be as if you could do it outside to actually study biodiversity. This is a zoo in rural England. It's Hamerton Zoo. Zoos are great for this because you know where all the animals are and you know exactly what they are. They're totally controlled, and they're not native. If you're in rural England and you find tiger DNA in the air, there's really only one source for it. So you know where they were and where it came from. So we walked around the zoo with our filtering systems, and we took samples from inside their sort of shelters of where the animals were, and then outside in the sort of free range land. So we're out in the environment where we want to be able to do this, but with a totally controlled set of animals to find. And we found them. So this is a top-down view of the zoo, like an aerial map. And each of those circles in the middle is, I have a pointer for this, sampling sites. And this is what we detected. So if you're in the gibbon enclosure, you find gibbons. And if you're in the binturong enclosure, you find binturongs. And if you're in the primate house, you've got lemurs and howler monkeys and tamarins and sloths. Great, things are where they're supposed to be. But you also find other stuff. Over in that primate house, you've got dingoes and donkeys and gibbons and budgies. This is DNA moving through the zoo. In fact, we could detect it up to 300 meters away from the source. The other great thing about the zoo is we know exactly how far it could possibly have gone between those dots. That's why we use zoos. And so we were able to detect other animals' DNA effectively in the wind. And this is great. It means you don't have to be right in front of the animal to detect it. You can detect it at a distance. And then we found wildlife. Muntjac deer are locally invasive, squirrels. What else have we got up here? Myotis bats, ducks, hedgehogs, locally critically protected species in Europe. And so we're seeing the wildlife. That was our end target. We already found them in the zoo, their DNA floating in from outside. And then we also found food. So over here, binturongs have a real preference for chicken, so we've got big spikes of chicken DNA. Whereas over here, we've got dingoes, they eat mostly mammals, so you've got pig, horse, and cow DNA. So we're seeing their food actually in the wind. Then over in Texas, another group in Texas was working on passive sampling for plant material in dust. So they have these marvelous traps. They actually have a sail on the back, so they will always turn into the wind, no matter which way it comes from. And they collect dust. And every 2 weeks, they come and wash the dust out, and they're looking for plant material. Well, they called us up and said, well, we never thought of looking for animals. Would you like to try? And so we did. And this is not as complicated as it looks. Each column is a sample across a whole year. So there's 22 samples across a 52-week year. And then each row is an animal we detected. And some interesting things. Okay, so these are bunny rabbits. And common things like bunny rabbits are commonly detected. This is good. This is a Texas toad. It's very rare to find the Texas toad out in the Texas rangeland. And great, it only appeared once. So rare things are rare. This is also good. There's a really weird thing that happens here. This whole time period, there's almost nothing detected. So green things detected, gray things missing. So what was weird about this time of the year? This is when there's hot, reduced vegetation. The animals basically go somewhere else. And when they're not there, we don't detect them. And in comparison, this is cooler, rainy, windy season. It's mating season in Texas. And they all suddenly get active. We begin to detect them. And the most dramatic case of this is that Texas toad. The Texas toad has some weird biology. Basically, it sleeps most of the year, and it will come out and mate quickly on an environmental cue, like the giant thunderstorms that happened the week before. So we had an environmental effect, caused an animal to become active, and we pick it up in the air. This is really good because this tells us that the eDNA detections in air samples are because there's activity. You won't want to detect something when it's not really there. You want to detect it around the time it was active for this to be useful. So following that, we built some samplers. They look like this. They're modified computer fans. They work like tiny vacuums. And if you live at my house, you get asked to help out. These are my kids building these things. That's my daughter testing out batteries. And if you're my niece and nephew, and it's break week from school, Aunt Beth makes you come out and sample things. So they're out testing these with some bird boxes in our local conservation area. And these things work. So this is a filter we built for siphoning DNA out of the air samples. It's always fun at my house on the weekends. And this is Belize. So in Belize, we go down and all the bats that we catch in the nets have to come into a classroom where I can identify them for you. That's my job while I'm there, is to check the names. Each of those bags holds a bat. That's how we hold them for brief periods of captivity. Those are my samplers, and there they are back there. And so we took these things down to Belize on one of our trips with a simple question. This is kind of like the zoo. I now know exactly how many bats of every species were in this room every single day. How good would I have been if I'd just taken an air sample? And the answer was amazing. We were about 90% accurate at telling you what was in that room over the course of 2 weeks. That's pretty good. Think of this like an artificial cave. We're bringing the animals in and out every day as if it's a bat cave. In they come, out they go, and it works. We also got other stuff. We found howler monkeys, toads, cats, chickens, peccaries, kinkajous. And we were thinking about this for a while. What did we do? This is post-COVID. First thing you do is you open all the windows and you put the fans on to keep their air circulation up, pulled all the air in from the outside. In comes the howler monkeys and the kinkajous into the room just from the air samples. So this is very, very good in this now sort of slightly more complicated. There's 40 different species of bat, 1,000 different individuals in that room. Some species only had one, others had hundreds, and we got them all, almost all of them. So then my student Nina decided to take these out to real caves. So this is Nina and Jono, and they're out deploying these, doing the first applied roost survey just by air. So in caves, this is pretty neat. This is actually Nina inside a tunnel cut by looters into a Mayan ruin. And the bats use the tunnels. So the people go in looking for artifacts, looters, and she's now inside sampling the bat communities just with air. Tree roosts, these amazing trees where the bats live inside them. There's another tunnel into a Mayan ruin. These are enormous. So that's Nina versus the tree, putting our samplers inside. And we already know from all of our surveys the kind of things we tend to find. There's a vampire bat, a nectar bat, a fruit bat, an insectivore. These are all things that we know live in some of these caves. We detected every single one just with an air sample. And the coolest thing was this thing. This is a white-winged vampire bat, so called because of the white tips on its wing. In 24 years of working in the tropics, I've never seen one alive. Our key to the area said, I wrote the key, there's probably white-winged vampire bats here. If you ever see one, make sure you bring it back for us to look at. First time out, the DNA was caught. So we've never seen one, but we have their DNA. Now that's what airborne eDNA is going to be cool for. Finding something we predicted was there based on the environment and the range, we confirmed it with DNA. We've never seen it with any other technique we have for catching bats because they're hard. So airborne DNA found missing species for us. It also found Coroosting. These are little Otolomies rats that live in the caves. We confirmed them with photographs and sound, and we now have their DNA. So airborne DNA is nice. We've got animal and plant DNA we know is now carried in the air. We can use passive collectors like the little thing with sails We can use my little vacuums for active collection. We've got really good evidence that it reflects recent activity and that we can accurately describe complex communities like a cave full of 1,000 bats. But we come back to the same problem. This is actually my first field site in Costa Rica. That's actually mist. With the lights on, you can't see it so well. The trees under here, mist rising off the forest. The problem is, when I was a student working in this forest, we struggled to describe its biodiversity. And I don't know how many years it's been, 25 years later, I don't think we're any better at it. It's a great, nice tool for surveying bats and their roosts, but it still doesn't solve this problem, this biodiversity problem. How do I measure that? That is what we need to be able to measure all the time, not once a year, constantly to monitor it. How do we do it? So the biodiversity problem, the third problem, is that we have no standard methods. Everything is taxon specific with some expert you need on site to do it. And because nothing is standard, we have no standard metrics for biodiversity. We have some vague ways of saying diversity is going up or down, but nothing really complex because we can't standardize it. Why? We haven't got any infrastructure to do it. We've got people like me with a net or a tiny little hand vacuum, but it doesn't scale. But we were wrong. We were totally wrong. Every scientist has written, we do not have global infrastructure. I think we're wrong. When our zoo study was done, there was an article published by the Natural History Museum in London. It was read by a physicist. And he called me, and he called my research partner, Joanne Littlefair. And he said, what is it you're trying to do? And so we explained about biodiversity declines and challenges, needing new tools and this. He said, you mean you just want to take samples of air? I said, yeah. He said, well, have you thought about the pollution monitoring networks? And we said, what are those? James Allerton works for the National Physical Laboratory of the UK. And they work on behalf of the Environment Agency through DEFRA. And they work under multi-country transnational agreements. And in his case, they're trying to monitor heavy metals, lead, arsenic, offshoots of cars. When you run your car and you burn gasoline, this stuff goes into the air. And for human health reasons, there are international standards on how many particles per cubic meter of air you're allowed. And in order to do that, you have to have networks of air quality samplers to measure that. And he runs one of those networks for the UK. This is one of the stations they have. It's a semi-automated system, which pumps a meter of air per hour through a filter like this. And after a week in the thing, it looks like this. And he said, well, is it possible we've been capturing DNA on these things without knowing it? Because the people who operate these are chemists and physicists. So he sent us some of these pieces after they'd done their analysis. We got 8 of them from Scotland. This is the Scottish Heathlands, is one of their stations. It's on the network. It takes a sample every week, over and over and over again. And every few months, someone goes and collects them all and brings them back for analysis. Over and over and over again. So they sent us some of those. This is a station outside Bushy Park, which is an urban forest on the edge of London, hugely biodiverse park. This is actually not on the network. So we were able to manipulate how long it sampled for, an hour and a day and a week. And we did that a few times. With the simple question, is it possible that these things are capturing eDNA out of the air? The answer is yes. 17 samples from 2 locations, 180 different taxonomic groups were represented on those filters. 80 different genera of plants, trees, garden plants, ornamentals, grasses, crops, all recorded in DNA signatures on a little filter piece. Fungi. 34 species of bird, 26 species of mammal, and a whole variety of arthropods and spiders and even a newt. Remember, we don't have any generalized sampling tools in biodiversity science. This is a complicated graph, and I'm going to delete most of it for you to show you the important bits. So this is the experiment for an hour and a day and a week outside of London. This is just the plants. And they show you something quite important about plants. After an hour, we found about 20 different genera of plants. And after a day, we had about that many, whatever that is, 35. And after a week, we really didn't have any different amount because plants don't move. They're there. After a day, you've got most of them in the area. You don't get many more in a week. If you contrast this to the vertebrates, these are the birds and the mammals. After an hour, you got a couple. After a day, you've got close to 20. After a week, you've got close to 30. Why is that going up? Because they move. The more that move into the area, the more we capture because they're mobile. This is about encounter. What's going to the area? Good evidence this is local. You aren't getting stuff blowing in from a long way away. It's how they have to go through that area. And the next important thing are these two. These are the ones from Scotland. And the reason they're important, so here's a week in London and a week in Scotland and a week in Scotland. And the reason those are really important is that we did not know at the time that when they sent us those Scottish samples, they had sat in a little shelf in some lab close to a year. And we couldn't tell the difference. The DNA hadn't degraded. It was there. And this becomes important in a minute. So this looks like a tool for measuring biodiversity. So now we've got a question. How far is it actually going to be able to travel? How long does a signal last? And how well does it match known distributions of species? And the UK turns out to be the absolute perfect place to have started this because they don't just have 1 or 2 sites. They have 150 sites that take absolutely identical measurements every week over and over and over again. Just in the UK, they are taking close to 8,000 absolutely standardized identical measurements. And we think they all caught biodiversity. And the UK is really very interesting because they also have an incredible knowledge of their own biodiversity, partly because so much of it has been lost already. They track what they have. And so they have these amazing maps. Here's the deer people. They can tell you to the square kilometer the approximate abundance of every species of deer. They've done this for virtually every plant. You can tell you across latitudes how many different plants are where and at what season. So you can take these maps and you can take, this is a map of the sample locations of just the heavy metal network. And we can compare what does this station measure and does it look like this deer and this deer, but not this one? So we can use these maps to validate those questions of where the signals come from. But this isn't a British thing. This is Europe. Every one of those stations is taking the same measurement because these agreements on human health and detection of pollutants are transnational. Everybody has to measure this way. We think every one of those dots is taking the same measurement. Here's you. There's your 1,000 sites taking absolutely identical measurements over and over. These are basically automated systems. Some countries, these have been operating since the 60s and the 70s, and some countries have never thrown them away. We could have, in some places in the world, decades of time series data on biodiversity captured as an accident, as a byproduct of something totally different. These are weather stations for planetary biodiversity, and they're everywhere. We found them in the Amazon. We found them in Australia, New Zealand, South Africa. They're everywhere. I now have maps like this from all over the place. They're not going to be perfect. Nothing in biodiversity is ever perfect for taking a measurement. But these are being collected whether we analyze them or not. We already have national systems to distribute the materials, collect them, centralize them, analyze them, archive them in some places. And we now know there's more to these samples than pollutants. When you hear about forest fires and dust and smoke in the air, it came from those. If you turn on your iPhone and go to your weather app and it gives you an air quality index measurement, it came from those. We already have systems to collect, regulate, analyze, and distribute this data. We just didn't realize that there was something else hidden on these. And I think that they all have DNA on them. So what we have is a globally distributed, semi-automated system collecting samples of what I think is terrestrial DNA every day, everywhere, all the time. This is global infrastructure for the measurement of planetary diversity. We don't need any infrastructure, it already exists, and it scales. I've gone from not having enough samples to no idea how to deal with that number. Somebody needs to build me factories to crunch through this stuff, but they're being produced and literally thrown away every single day because nobody knew there was added value to them. Okay? This is something that can scale. This is something that can measure biodiversity on a national, continental, or potentially global scale, and we are finding them everywhere. Most universities have one on the roof for the chemists. They're not part of national grids, they're just there. Private industries have them. In Canada, every mine has one because they have to monitor their particulate matter they put into the atmosphere from mining, just there, capturing like this all the time. So I'm going to go back to this. So as I said, I'm part of Bioscan, and the Bioscan mission is on one side to build the reference libraries of genetic information so that someone like me can come through and sequence off one of these filters with unknown sequences and match to a database and figure out what those unknowns are. That's what we do. It's like the kids' card game, Go Fish. I've got one of these. Have you got one to match me? That's how we do it. Unknown sequence, does it match what you've got in your database? If it does, I can identify it. That's how it works. It's not much more complicated than that. So Bioscan is trying to build the reference databases. It's also trying to come up with a way of surveying diversity and the changing diversity over time. And I suspect that these networks of air quality samplers might be the key to doing that, to actually answering the conventional and biological diversity and being able to measure things. Could we just detect an invasive species as it arrives from an air filter? Could we find a range shift as a group of animals moves north because of warming? Can we figure out whether populations are in steep decline or increasing? Maybe. I'm going to try, if I can, to leave you with a video recreated about Bioscan, just to give an idea of what we're trying to actually accomplish. Maybe.
Our living planet, a library of life. Every species like a book, holding the information for humanity's greatest innovations. New medicines, technologies, and economic development. But we are burning those books before we've even read them. We want to know all life, every species. We are the International Barcode of Life Consortium, and we are illuminating biodiversity through DNA-based analysis. By using automation and rapidly evolving DNA sequencing technology, we are bringing this vision to life. Our new scientific program, Bioscan, is shedding light on unknown species, tracking their distribution, abundance, and interactions. Bioscan will bring us closer to building a complete library of life, forming the basis for an Earth observation system. Together, we must act. We are not just scientists. We are rangers in national parks collecting the data required to monitor ecosystems. We are Indigenous peoples protecting our lands and traditions for future generations. We are citizens supporting science and conservation in our country. This is Bioscan, collecting, mapping, and revealing life's diversity, our species acting to protect all species before we lose the possibility of even knowing them.
Please join me in thanking Professor Clare again for an inspirational talk. I think she has a lot of work ahead of her by the sounds of things. And it would be great. It's great to see you all again. It'll be great if you join us again next month when we have to talk about human evolution. Thanks. Safe home.